Hello students, we will talk about in this lecture bioassay of insulin, oxytocin and vasopressin. In our last lecture we had talked about bioassays, how they are carried out, what are the types of bioassays and how these bioassays could be performed. I am Dr. Monica Sachdeva, professor at Rajkumar Goyal Institute of Technology. This presentation, this lecture number 43, this will take you through what is insulin, what is the use of insulin and what are the various methods by which bioassay of insulin could be performed. Then what is oxytocin, what is the use of oxytocin and how the bioassay of oxytocin could be carried out. Then we will also talk about bioassay of vasopressin. What is vasopressin? What is its used? How it is released? How it is controlled by the hypothalamus and how bioassay of vasopressin could be carried out. So let us start with our lecture. We will first talk about bioassay of insulin. Before talking about how this bioassay of insulin is carried out, we will just go through quickly what is insulin. We all know that insulin, it is a pancreatic hormone and it is released from the beta cells of pancreatic beta cells. And the function of insulin is that it controls the blood glucose level or blood sugar level by uptake of glucose and converting the glucose into glycogen. And insulin is used in type 1 diabetes, which we call as Insulin Dependent Diabetes Mellitus. So let us now start with bioassay of insulin. We talked about that whenever a bioassay is performed, you have to have one test sample and another internationally approved standard sample. So that is compared, the test sample, the potency of the test sample is compared with the internationally accepted standard sample. And all the bioassays, since we are carrying it on the animals, I will once again repeat that all these animal experimentation that has to be approved by Institutional Animal Ethics Committee, which we call as IAEC, which is under CPC-SEA. So let us start with this. What are the standard preparations and units of insulin? Insulin is a pure, dry and crystalline insulin, and 1 unit of insulin, it contains 0.04082 milligrams, and this unit is specified by Ministry of Health, Government of India, and this is equivalent to 1 international unit. So 0.04082 milligram, this is 1 international unit of insulin. Now how do we prepare this standard solution? To prepare standard solution of insulin, you have to accurately weigh about 20 units of insulin, dissolve it in normal saline, and please remember that 1 unit is equal to 0.04082 milligrams. Then 20 units dissolved in normal saline. This is acidified with HCl. pH is adjusted to 2.5. To this it is added 0.5% phenol, which is used as a preservative. To this sample, in a glycerine in the range of 1.4% to 1.8% is added, and the final volume which we make up, that contains 20 units per ml of the solution. So this is the standard solution, 20 units per ml. And we have to store this solution in a cool place, and this could be used, this standard solution could be used within 6 months. Now, we also have to have a test sample, and the test sample is also prepared in the similar way as we had prepared the standard sample. Now, to carry out insulin, bioassay of insulin, we have various models or methods, we can say, by which bioassay of insulin it could be performed. These are the rabbit model, where we observe the hypoglycemic effect of insulin. Then is the mouse model, where we observe the hypoglycemic effect and also the hypoglycemic convulsions which are recorded while we are carrying out the bioassay of insulin. Then is the isolated rat diaphragm model. In this, we kill the rat, we take out the diaphragm, homogenize it and calculate the amount of glycogen which is present in the diaphragm, because the one of the action of insulin is to convert glucose into glycogen. And then another model is rat epididymal fat model, where we observe the glucose metabolism when we administer insulin. So, we will be talking about in detail about rabbit model and the mouse model. Now, what is this rabbit model and how this bioassay is carried out? Now, since we are using rabbit, so the first and the foremost thing is that we have to select the rabbits. Now, while selecting the rabbits, it should be seen that all the animals, they are healthy and they weigh about 1.8 kilograms to 3 kilograms. The diet which we are putting on them on, that should be uniform. The pellet diet or the diet which is recommended by the vet, it has to be followed and it should be maintained constant throughout your bioassay. And when we have to perform the bioassay, 18 hours before the performing bioassay, we have to keep the animals on fasting. And water is also withdrawn during the experiment, but when we are keeping animals on fasting state, the water is ad libitum is allowed. Now, we have the standard sample, we have the test sample also. We make certain dilutions of the standard and the test samples by diluting them with normal saline solution or sodium chloride solution, and so that the final standard and the test dilutions, they have 1 unit per ml of solution and 2 unit per ml of solution. So, we have 2 standards, 1 unit per ml and 2 unit per ml, and we have 2 tests, 1 unit per ml and 2 unit per ml. And the principle of this bioassay of insulin is that we carry out or we estimate the potency of a test sample by comparing the hypoglycemic effect of the sample with that of the standard preparation of the insulin, because insulin का action है that it causes the hypoglycemic effect, that is, it lowers the blood glucose level. So, this is the principle that the sample and the test samples का जो hypoglycemic effect record होगा, that we have to accordingly we have to compare both the test and the sample and carry out or estimate the potency of your test sample. Now, how this is done? What is the experimental procedure for the rabbit model? Now, these animals, they are divided into 4 groups. We take 3 rabbits in each group. This experimental procedure has got 2 parts. The first part of the test is where we measure the initial blood sugar level. Now, whenever blood is to be collected from the rabbit, we use the marginal ear vein of the rabbit. If you have observed the rabbits का जो ear है, it is very thin. उसमें veins जो हैं, they are very clear. So blood is withdrawn from those marginal ear veins. Then we calculate the reducing sugars. It is estimated per 100 ml of the blood by using a suitable chemical method, and this we call as the initial blood sugar level. Initial means जो fasting blood sugar level था, we have calculated. Now we have 4 groups, 2 test and 2 standard test. So group 1 we take standard, treatment दिया हमने उसको standard 1. We have group 2 which we call as standard 2, treatment दिया हमने उसको standard 2 का, test 1 को test 1 दिया और test 2 को we give test 2 sample. Okay, so all these injections of insulin, they are administered by the subcutaneous route. Then after administering insulin, after every 5 hours, I mean up to every 5 hours, every 1 hour के बाद में we again withdraw the blood sample and again estimate the blood sugar level, and this we call as the final blood sugar level. So first part is where 1 we carry out the fasting level, initial blood sugar level, then we divide the animals into we have the division of the animals into 4 groups. We administer them the subcutaneous insulin and determine the blood sugar level for 5 hours every 1 hour, and we call that as the final blood sugar level. Then the second part of the test is we call as the crossover design. In the first part, what we carried out was a parallel study. Standard 1 animal को we gave standard 1 treatment, standard 2 को we gave standard 2 treatment, test 1 treatment animal को we gave test 1 and test 2, so on. But in this group, what we do is जिसको हमने भी standard दिया था animal को, we now give it the test, और जिसको हमने lower dose दिया था standard का, उसको हम higher dose of test देंगे. Fine. So if see here we have done what we have done is that the standard 1 animal, it now receives test 2. Otherwise, in earlier first part it was receiving standard 1. Now standard 2 को we give the test 1, the lower dose, test 1 we give the standard 2, and test 2, this is test 2, we give it the standard 1. So after same initial blood sugar level and final blood sugar level is estimated by chemical method, and the mean percentage decrease in blood sugar of the first part and the second part is calculated. So this gives you an idea of the hypoglycemic effect, whether your test sample, what is the potency of your test sample in comparison to your standard, whether जो हम नया drug जो हम लेना चाह रहे हैं क्या वो insulin से compared है या नहीं है. So this will decide. This is called as the rabbit model. I'll once repeat it once again, that for the rabbit model we have first part of the test, initial blood sugar level, we give subcutaneous injection of insulin, calculate the final blood sugar level, calculate the mean. Then the second part, the crossover design, the animal which was receiving standard now receives the test, animal which was receiving higher dose of standard now receives the lower dose of the test. These are the crossover tests. You calculate the mean of all hypoglycemic activity or decrease in the blood sugar level. This is how the bioassay of insulin is carried out. Then you have a mouse method. Now in this, the principle of this mouse method is when we give insulin to the mouse by subcutaneous route, at certain elevated temperature, the convulsions are produced, which we call as the hypoglycemic convulsions. And this percentage of hypoglycemic convulsions which are produced by the test sample, they are compared with that of the standard preparations. The procedure for preparation of the standard sample as well as the test sample, that remains the same, which we had discussed earlier. Now, in this case, the animals, around 100 mice are used. You can use albino mice, Swiss strain mice can be used. The weight should be around 18 to 22 grams, but all the animals should be of same strain. They should be maintained at the constant diet. Now, the dilutions which we are doing, the standard dilution 1, it is 0.064 unit per ml, and the standard dilution 2 or the second sample, it contains 0.096 units per ml of the solution. Same test solutions are also prepared. Now, the experimental procedure, it remains the same. We have same 4 groups with 25 animals each. Initial, we give the subcutaneous injection of insulin, and after giving the insulin, with all the mice, they are put in an air incubator at 33 degrees centigrade, and they are observed for 1.5 hours, and the mice which show the convulsion or die, they are then taken out and they are observed. Okay, and the percentage convulsions which are produced by the test sample and the standard sample, they are compared, and that is how this bioassay of insulin, it is carried out by using mouse method. We will now talk about bioassay of oxytocin. Now, before moving on to how do we carry out bioassay of oxytocin, we will first talk about what is oxytocin and where it is used. Oxy means rapid and tocos means rapid and oxy means life. So, oxytocin means it is a polypeptide hormone which is released from the posterior pituitary, and it stimulates the contraction of uterine smooth muscles and also the mammary gland. So, while parturition or while delivery of a child, this oxytocin is used to stimulate the uterine contractions and also ejection of the milk from the mammary gland. So, the physiological role of oxytocin, it causes the contraction of the uterus during labor and also causes milk ejection. It is also known as the love or the bonding hormone, and it also promotes the feeling of well-being. Now, when we carry out the bioassay of oxytocin, same the potency of a standard oxytocin is compared to that of the test oxytocin by various methods which we will be using. The standard preparation of oxytocin was established in 1978, which is now an accepted standard, which is a freeze-dried synthetic oxytocin peptide with human albumin and citric acid, and this is supplied in the ampoules which contain around 12.5 units. This is the standard acceptable unit of oxytocin. Now, we use various experimental methods. We use it by depression of blood pressure in chicken. We can also use the contraction of rat uterus, and we can also measure the test sample. We can carry out bioassay of oxytocin by measuring the milk ejection pressure in a lactating rat. We will talk about method A and B to carry out oxytocin bioassay. Now, the method A, it is depression of blood pressure, and this is carried out in chicken. In this case, young male chicken which are around of 1.2 to 3 kg, they are used. They are anesthetized so that the animals or the chicken, they can be maintained on a constant high blood pressure. Then the gluteus primus muscle, which is also called as the thigh muscle, this is exposed. We locate the popliteal artery and also the brachial or the crural vein. Popliteal artery is cannulated for monitoring of BP, and the brachial vein for infusion of your test or the standard solution. So we prepared the standard solution with the saline, inject 0.1 to 0.5 ml of the first dose of standard solution into the cannulated vein and record BP. Dose, it should cause decrease in the blood pressure, and the required dose, it should be between 20 to 100 milliunits of oxytocin. Now after an interval of 3 to 10 minutes, or you can say the rate at which the BP returns to normal of the first dose, you can administer the second dose, and again the BP is recorded. So in this way, the ratio of the test sample and the standard sample, that remains the same, and you have to take around 6 responses of the standard as well as of the test, and then the fall in the blood pressure with your standard sample and with your test sample, they are compared, and potency of your test sample is then carried out. You apply various statistical methods, and the potency is calculated. Then another method is contraction of the rat uterus to determine the bioassay of oxytocin. Now in this case, female rats are used, which weigh around 120 to 200 grams. Generally, Wistar strain rats are used. Now we have to see it here when we are carrying out the bioassay of oxytocin on uterus. We need to see that all the animals, they are in the estrus cycle. Fine, if the rats, they are not in the estrus cycle, then we have to inject them the estradiol benzoate is administered to them by intramuscular route, and the vaginal smear is then taken out is to see in which cycle, which estrus phase the rat is. And again we prepare a solution, a physiological salt solution, dilution, which consists of various chemicals and various minerals. We have to maintain the temperature at 32 degrees C. We provide oxygen, we provide aeration to the animal, and we use the isotonic lever, and the contractions of the test sample and the standard sample, they are recorded. We can carry out this bioassay by means of either bracketing method, we can also carry out by means of graphical method, or any of the multiple point bioassay method. So here the sample will be your rat uterus. We have to isolate that rat uterus in vitro experiment, graded bioassay method. We take out the uterus, put it in a physiological salt solution, which is dilution. You give aeration, maintain the pH, maintain the temperature. All external conditions, they are provided, all internal conditions are provided externally, and we carry out the bioassay and find out the potency of your test sample in comparison to the standard by using any of the method. This has to be repeated by the 6 times. All these bioassays, they have to be repeated. Then we talk about the bioassay of vasopressin. Now what is this vasopressin? Vasopressin is a peptide hormone, and it is secreted by posterior lobe of the pituitary gland. Now this has a vasopressor action. It can stimulate the contraction of arteries and the capillaries, hence it is named as vasopressin, and another name for it is antidiuretic hormone, used in case of diabetes insipidus. See here that the hypothalamus, whenever there is a decrease in the blood pressure, its hypothalamus stimulates the posterior pituitary, it releases vasopressin, and vasopressin, it acts on your blood vessel and the kidneys, increases blood volume, increases the resistance and increases the blood pressure. Now, to carry out the vasopressin, again the standard sample and the test sample, two samples are there, and you have to find out the potency of the test in comparison to the standard. Now, your standard preparation is a dried acetone extract of the posterior lobes of pituitary gland of oxal, and the standard unit is that the specific pressure activity corresponding to that yielded by 0.0005 grams of standard preparation, which comes to around 20 units per ml. This is the standard preparation, and the experimental models we carry it out by the rat model and the cat model. Now, here the albino rats are used. We anesthetize it by using the subcutaneous injection of ethyl carbamate. We cannulate the trachea for artificial respiration. Then we cannulate our carotid artery for BP monitoring. Femoral vein is also cannulated for drug administration. Heparin is injected so that no coagulation takes place. Hypotensive unit is given into the tail vein so as to maintain a constant blood pressure. And then your sample preparation and your test sample, they are administered. The volume should be around 0.1 to 0.5 ml, and the decrease in the blood pressure with both the standard and the test sample is recorded, and the potency of the test sample in comparison to your standard sample is then calculated. The fall in the blood pressure, that is then carried out. So, this is record in the blood pressure response, that is then carried out. The rise in the blood pressure is carried out, and then the mean test results, they are then carried out with the standard. Similarly, the cat model can be used, same procedure, the only here we use the adult rats. We anesthetize it, cannulate the artery, cannulate the vein, maintain the normal blood pressure, administer the doses of the standard and the test, and record the maximum rise in blood pressure in response to each dose, which is the physiological action of your vasopressin. And then your mean test results, they are then compared with the standard. So, that is all. We talked about the bioassay of insulin, where we use the rabbit model and the mouse model hypoglycemic convulsions. Then we talked about bioassay of oxytocin. We also talked about bioassay of vasopressin, where increase in the blood pressure response with both the test and the sample is recorded. So, that is all about this lecture. Thank you.